say, Teacher Roby. Now that we are in a new normal, most of us are staying at home. Because of this, we tend to do a lot of activities in order to make use of our time. It can be cleaning our house, doing household chores, cooking our favorite food, and exercising. Now, in order to do these activities, we need to have a lot of energy. Now, have you ever wondered how the food that we eat is converted into energy? Now, if you want to find out the answer to this question, please tune in this video lesson.
This video lesson for grade 9 biology is about ecosystem life energy. The specific lesson that we are going to discuss is about cellular respiration. When people and other heterotrophic organisms eat food from producers and consumers, chemical energy stored from food is transferred to their bodies. Do you know how these processes are being done by our body and by other living organisms? How do cells convert stored energy in food into chemical energy? And how do materials and energy flow in the ecosystem? Now, if you want to know the answers to these questions, tune in this video lesson. Let us try this pre-assessment for us to find out how much you know about the cellular respiration. Directions: identify what is asked or described in each item. Choose the letter of the correct answer. Number 1. All organisms get energy from food to perform different life processes. This is done in the cells by breaking down sugar molecules into chemical energy. Which of the following cell organelles is associated with the production of chemical energy? A. Chloroplast, B. Mitochondrion, C. Nucleus, D. Endoplasmic reticulum. Number 2. Pyruvate is the product of glycolysis. If there is no oxygen available to cells of the human body, what becomes of pyruvate? A. Alcohol, B. Lactic acid, C. Carbon dioxide, D. A and C. Number 3. If you did not eat for 3 days, where did your cell get the glucose for ATP production? A. Blood sugar, B. Protein in the blood, C. Glycogen in the liver, D. Glycogen present in the muscle. Number 4. How many molecules of carbon dioxide is or are released from one pyruvic acid molecule being oxidized? A1, B2, C3, D4. Number 5. During the synthesis of ATP, what is the direction of hydrogen flow? A. From intermembrane space to matrix, B. From matrix to intermembrane space, C. From cytoplasm to matrix of mitochondrion, D. From matrix of mitochondrion to cytoplasm. How did you find the pre-assessment? Did you find the quiz difficult? No worries! You will learn more along the way, so let us get going! Are you curious already what cellular respiration is? Now we have here a diagram that summarizes the processes of cellular respiration. As you can see, it is divided into 3: glycolysis, Krebs cycle, and electron transport chain. You are going to know more about these processes as we move on with our discussion. Let us discuss first cellular respiration. Cellular respiration is catabolic. This means organic compounds are broken down into smaller molecules like carbon dioxide and water, and energy is released. Carbohydrates, mainly glucose, and fats are the main sources of energy. Proteins are rarely used for energy. They are important as building blocks for growth and repair. There are two types of respiration. First is aerobic, called biological oxidation. It needs oxygen. In simplified version, we have C6, H12O6, and 6O2 react in the presence of enzymes to produce 6H2O, 6CO2, and energy. Second is anaerobic, called fermentation. It doesn't need oxygen. The overall process is simpler and faster than biological oxidation, but produces much less energy. Now, I want you to do this task. Open a new tab for YouTube and type the link that is projected on your screen. You can jot down some notes and enjoy watching. And to unlock the difficult words that you are going to encounter in this lesson, we have here the word bank. Cellular respiration is the process by which energy is harvested from food. The breakdown of glucose involves three major steps: glycolysis, Krebs cycle, and electron transport. Glycolysis, the first step of cellular respiration that occurs in the cytoplasm of the cell. During this stage, the 6-carbon glucose C6H12O6 is broken down into 2 molecules of 3 carbon sugars, glyceraldehyde phosphate or GAP, and dihydroxyacetone phosphate or DHAP. Krebs cycle occurs in the mitochondrial matrix and generates a pool of chemical energy, ATP, NADH, FADH2, from the oxidation of pyruvate, the end product of glycolysis. Electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria to form a gradient of protons that drives the creation of adenosine triphosphate or ATP. Let us explore more about cellular respiration in this activity. This is entitled Cellular Respiration in Yeast. The materials that you will be needing are the following: distilled water, 2.5 grams table sugar, baker's yeast, balloon, pH indicator, test tube rock, and rubber band. For the procedure, you are going to do the following. First, warm water to 37 degrees Celsius. Second, place lukewarm water in a test tube. Third, add sugar to produce 5% sugar solution. Dip a pH paper into the mixture. Match the color of the pH paper with the accompanying color chart. Fourth, drop 5 to 10 granules of yeast. Fifth, mix with swirling motion. Sixth, place the test tube in a test tube rack. Seventh, attach a balloon to the mouth of the test tube. Eighth, secure the balloon with a rubber band. 9th, wait for a few minutes. 10th, once the balloon is fully inflated, remove the balloon and tie it with a rubber band. 11th, dip in another pH paper. Note the color change of the pH paper.
This time, learners, I will be performing the activity entitled Cellular Respiration in Yeast. The materials that we will be needing for this activity are the following: distilled water, test tube rock, pH indicator. As for me, I will be using a litmus paper with color chart, balloons, rubber bands, 2.5 grams of sugar, and baker's yeast. So come on, let's start the activity. I have prepared already the lukewarm water at 37 degrees Celsius. This time, we are going to place the lukewarm water in a test tube. Make sure not to fill the test tube since we are going to add some sugar to it. Okay, this time, we will be adding the sugar. We need to have a 5% solution for the sugar. And then afterwards, using a litmus paper, we are going to dip this one in the mixture. Kindly observe for any changes in color in the litmus paper. Afterwards, using a pH chart, you are going to compare the litmus paper that you have dipped in the mixture with the standard color found in the chart. Do you now have your results? Kindly write it down in your notebook. This time, using the mixture that we have prepared earlier, we are going to add 5 to 10 granules of yeast. We are going to mix it in a swirling motion. Once all the granules have settled, we are going to place the test tube in a test tube rock, and we are going to attach a balloon to the mouth of the test tube. And in order to secure the attachment, we are going to tie it with a rubber band. And then, we are going to wait for the balloon to be inflated. Once the balloon has fully inflated, we are going to remove it from the test tube. And then, we are going to use another litmus paper and we are going to dip it in the mixture. Afterwards, still using a color chart, you are going to compare the result in the litmus paper in the standard color. And now, you can write down the results and answer the guide questions that will be projected on your screen.
These are the guide questions that you are going to answer. Number 1. What does the yeast represent? Number 2. What is the purpose of sugar? 3. Why did we use lukewarm water? 4. What was the pH of the sugar solution? 5. What did you notice at the surface of the sugar solution when you dropped the yeast? 6. What happened to the balloon after attaching it to the mouth of the test tube? 7. What filled up the balloon? Explain your observation. I will be giving you 5 minutes to answer the questions in your activity notebook, so kindly set your timers. In the activity, you saw bubbles coming out from the mixture. This means that the yeast fed on the sugar. One evidence is the formation of bubbles. The gas in the bubbles is carbon dioxide. The mixture became acidic as shown by the color change of the pH paper. This time, we are going to discuss the 3 processes of cellular respiration. Let us start with glycolysis. We have here a diagram that summarizes the processes involved in glycolysis. Glycolysis is the first step of both aerobic and anaerobic respiration. It occurs in the cell's cytosol. It doesn't need oxygen, but it does require energy, specifically 2 ATP. Let us discuss the step-by-step processes of glycolysis cycle. Step 1. Energy is required to begin the process. So, a molecule of glucose accepts 2 high-energy phosphate groups from 2 ATP molecules. Step 2. The resulting intermediary molecule immediately divides into 2. 3 carbon molecules called pigal, each containing a high-energy phosphate group. Step 3. A second high-energy phosphate group is added to the 3 carbon pigal molecule and 2 NOD-H plus H plus molecules are produced. Step 4. Finally, the 3 carbon pigal molecules donate their high-energy phosphate to create ATP, and the 3 carbon pyruvate forms as the final products. 4 ATP molecules are produced in glycolysis, but since 2 are required to start the process, the net gain is 2 ATP. This is not sufficient energy to maintain complex life systems. So, pyruvate, also called pyruvic acid, which still contains energy, will continue into biological oxidation or fermentation, depending on the availability of oxygen. If oxygen is present, then pyruvate enters the mitochondria. The next process that we are going to discuss is Krebs cycle. But before we proceed with the discussion about Krebs cycle, I want you to try this activity. Your task is to arrange the following events in the Krebs cycle in proper sequence. Assign numbers 1 to 7 in the space provided or write it in your activity notebook. Just keep your answers because it will be checked by your science teacher. Now, let us get to know the second process of cellular respiration called Krebs cycle. The Krebs cycle occurs in the mitochondrial matrix and generates a pool of chemical energy, ATP, NOD-H, and FADH2, from the oxidation of pyruvate, the end product of glycolysis. The citric acid cycle, also known as the Krebs cycle or the tricarboxylic acid cycle, is at the center of cellular metabolism, playing a starring role in both the process of energy production and biosynthesis. It finishes the sugar-breaking job started in glycolysis and fuels the production of ATP in the process. Pyruvate is transported into the mitochondria and loses carbon dioxide to form acetyl-CoA, a 2-carbon molecule. When acetyl-CoA is oxidized to carbon dioxide in the Krebs cycle, chemical energy is released and captured in the form of NADH, FADH2, and ATP. And now we are down to the last part of the process of cellular respiration called electron transport system or electron transport chain. The electron transport chain allows the release of the large amount of chemical energy stored in NAD+ or NADH and reduced FAD or FADH2. The energy released is captured in a form of ATP, 3 ATP per NADH and 2 ATP per FADH2. The electron transport chain is a series of complexes that transfer electrons from electron donors to electron acceptors via redox reactions and couples this electron transfer with the transfer of protons across a membrane. The electron transport chain is built up of peptides, enzymes, and other molecules. Remember these points about electron transport chain. 1. Proton pumps create an electrochemical gradient or proton motive force. 2. ATP synthase uses the subsequent diffusion of protons or chemiosmosis to synthesize ATP. 3. Oxygen accepts electrons and protons to form water. For the reflection, I want you to complete the following sentence prompts. I have learned that. I wish to ask my teacher about. You can write your answers in your activity notebook. We are now done with our discussion about the 3 processes of cellular respiration. This time, we are going to assess what you have learned. Let us find out whether you have learned something from our discussion. Directions: identify what is asked or described in each item. Choose the letter of the correct answer and write it in your activity notebook. Number 1. Which of the following is the cell's immediate source of energy? A. Fats, B. Glucose, C. Amino acids, D. Carbon dioxide. Number 2. Where does glycolysis occur in the cell? A. Cytoplasm, B. Lysosome, C. Mitochondrion, D. Nucleus. Number 3. What is the product of glycolysis? A. Pyruvate, B. Carbon dioxide, C. Phosphoglyceraldehyde, D. Dihydroxyacetone phosphate. Number 4. How many ATP molecules are used up during glycolysis? A1, B2, C3, D4. Number 5. How many ATPs are produced during glycolysis? A1, B2, C3, D4. Number 6. What is the net gain of ATP during glycolysis? A1, B2, C3, D4. Number 7. Where does the oxidation of pyruvic acid occur in human cells? A. Cytoplasm, B. Mitochondrion, C. Nucleus, D. Plasma membrane. Number 8. When acetyl-CoA enters the Krebs cycle, what substance must it interact with? A. Citric acid, B. Malic acid, C. Succinic acid, D. Oxaloacetic acid. Number 9. In one round of Krebs cycle, how many carbon dioxide molecules are released? A1, B2, C3, D4. Number 10. In one round of Krebs cycle, how many NADH molecules are produced? A0, B1, C2, D3. That's it, grade 9 learners, for our lesson about cellular respiration. Congratulations for finishing this video lesson. Reminder, do not forget to keep your activity notebook because it will be checked by your science teacher. Contact your science teacher once you are done with all the activities.
Congratulations for finishing this video lesson. I hope you have learned something about cellular respiration. We still have a lot of video lessons coming, so please do not forget to tune in for more videos.